To begin, let us acknowledge that Queen's University is situated on traditional Anishinaabe and Haudenosaunee territory. We are grateful to be able to live, learn, and play on these lands. We thank the Indigenous peoples of this area for their care of this land for thousands of years, and we hope to honor and respect them during our virtual event today. I'd also like to take a moment to thank everyone for joining us today. Our speaker this afternoon is Alexandra Foley-Eby, who will be presenting on evaluable inheritance. Co-infection with multiple strains in the mother reduces the protective efficacy of the maternal antibody response in the offspring. Alexandra completed her Bachelor of Science in Biology at Dalhousie University in Nova Scotia, where in her 4th year, she completed an honors project with Dr. Tatiana Rosso-Limo on the prevalence of Rickettsia bartonella species in the local flea population. Fleas were a bit of an oddity in the lab, which generally focused on ticks and Lyme disease. And it was because of this connection that she pursued a Master of Science in Biology at Mount Allison University in Sackville, New Brunswick, under the supervision of Dr. Yvette Lloyd. Dr. Lloyd is renowned on her research on ticks and Lyme disease, as well as being a passionate supporter of patient-driven research. In her lab, Alexandra studied the prevalence of canine Borrelia infections on Prince Edward Island, and using this data to estimate human prevalence and potential underreporting of the disease. After her master's degree, Alexandra worked for Dr. Lloyd as a copy editor and laboratory assistant, and also for a private tick testing company named Gene Ticks as the director of operations in Atlantic Canada. In 2020, she made the decision to return back to school to complete her PhD at the Western College of Veterinary Medicine at the University of Saskatchewan in Saskatoon. For her PhD, she is supervised by Dr. Martin Vordeau, whose primary interest lies in the ecology of the Lyme disease bacterium and the biotic and abiotic factors that contribute to strain composition. In this position, Alexandra has been able to combine her interests in veterinary medicine and the interests of her supervisor to both answer and raise questions regarding the role of maternal antibodies in the ecology of Borrelia. We will have Alexandra present, and then we will open for questions from the audience. You can ask your questions by entering your questions into the chat box, or you can raise your hand or unmic. Please help us welcome Alexandra to the podium.
There, I'm unmuted now. Hi, everybody. So I'm just going to share my screen right now. Okay. All right. So my title is Invaluable Inheritance, Co-infection with multiple strains in the mother reduces the protective efficacy of the maternal antibody response in the offspring. You know my name, I'm Alex. My supervisor is Dr. Martin Vordeau, and I also have a co-supervisor who is Dr. Janet Hill. So a little bit about me. I'm originally from Toronto. I have my BSc in honors from Dal, that was mentioned. I have my master's from Mount A. I have experience working at Mount A with local patient advocacy groups. I also have that experience working for genetics, doing private tick testing. And I decided at some point that I wasn't done with school. So I went back to do my PhD, and I'm now just finishing my 2nd year. So I'm halfway through at the WCVM in Saskatoon. My end goal, if anybody's interested, is to do the DVM program at Guelph and then do veterinary research on Lyme disease in the future. Okay, so I feel like everybody who's here is probably pretty aware of what Lyme is, but I would be wrong to not give some background. So I'm going to go over that right now. Okay, so we're going to talk about cause, vectors, and reservoirs. So Borrelia is a genus of bacteria that are spirochetes. They include about 20 genuspecies, and they can be divided into 2 main groups. So what you're seeing here, and I'm just going to bring up my pointer. There we go. So up here, these are what are called the relapsing fever group bacteria. And down here are the Lyme borreliosis bacteria. And mostly that's grouped based on the symptoms of their respective infections. The species that you're probably familiar with in the LB or Lyme borreliosis group would be Borrelia burgdorferi, Borrelia fazellii. And if you're thinking about the relapsing fever group, species that's gotten a lot of attention recently is Borrelia miyamotoi. We're going to be focusing today mostly on Borrelia burgdorferi, and that's what I'm going to talk about right now. So Borrelia burgdorferi is tricky, especially for people who aren't from the Lyme community, because you're used to seeing a genus and species, and that refers to that species. But of course, you can say Borrelia burgdorferi and actually mean all of the LB group, in which case it would be referred to as sensu lato, which means in the broadest sense. If it's used to describe just the species in the classical sense, then it would be sensu stricto. I'm really going to be talking about sensu stricto today, unless I specify otherwise. Now, Borrelia burgdorferi sensu stricto is the primary cause of Lyme disease in North America. So how does Borrelia get into you? Because it's not airborne, it's not in the water. How are you getting it? Well, it's being transferred to you through the bite of a tick vector. And generally, those are Ixodes species ticks. And in Canada, we have two species that we know carry and transmit, which are Ixodes pacificus, which, as the name would suggest, has a more westerly distribution. And then there's Ixodes scapularis, which has a more easterly distribution. Ixodes scapularis is what I'm working with and what I'm going to be talking about today. There are 4 life stages to a tick. They are born as eggs. They are then hatching into larvae. Those larvae then need a blood meal to be able to molt into nymphs. And then those nymphs need a blood meal to molt into adults. And then the adult females need a blood meal in order to fuel their egg production, essentially. And I've tried several times to make my own tick life cycle, but it never looks as nice as the CDC one, so I always end up using it. I also really enjoy that this shows you how it is a multi-year process. So it's not something that's accomplished over a single year. And so if a lot of times, especially when I was working on PEI, there were a lot of questions about overwintering and why that matters. And so that's why it matters, is because it's a multi-year process. Hosts. So hosts are important for the tick and they're important for the bacteria. The tick needs to eat. And so generally, the smaller the tick, the smaller the host, although they're fairly indiscriminate. And when we're talking about reservoir hosts, we're really talking about what is a good host for the bacterium. So a good reservoir host is one that can be infected, so it doesn't fight off the infection. It maintains the infection and it doesn't succumb to it, because you're not very useful if you're dead. And it can actually transmit that infection to a novel tick. So remember me saying that ticks are born as eggs. Well, when they're born, when they're laid, they hatch into larvae with no Borrelia. There's no vertical transmission from the mother to the larva. So when a female is infected, that doesn't get transmitted to the offspring. So for a tick to become infected, it has to feed on an infected host. And that means that you need these reservoirs in nature to be tapped into by the next generation of ticks. Quite often, the best reservoirs are small mammals and birds. So think rodents, shrews. And then, of course, people always ask about deer, because they're, the colloquial name is the deer tick. Deer are a preferred food for adult scapularis, and my reading has suggested that they like to mate on the deer, but they're not good reservoirs for the bacteria. They're just a good food source for the tick. You have to remember that those are essentially two separate entities. And then, of course, humans are important because we are human, and they are really incidental hosts, but unfortunately, it can be pretty catastrophic when they're infected. So when we become infected and develop Lyme borreliosis, it's also called Lyme disease. It can look a bunch of different ways, and essentially the symptoms are related to the degree of dissemination of the spirochetes through the body. The characteristic rash that people like to focus on, and I've included a picture of here, is found in about 70 to 80% of infected persons. And that number varies depending on your source as well. This one is from the CDC. But the other symptoms, so if you're unlucky and you don't have an EM rash, you have symptoms that are very generic. So fevers, chills, swollen lymph nodes, arthralgia, meaning joint pain, myalgia, meaning muscle pain. And so if you're unfortunate and you didn't notice that you got bitten and you didn't either see the rash or you didn't develop a rash, it can be really hard to know that you have Lyme. If the disease progresses and is not treated, then you can develop other problems as the spirochetes travel to different parts of the body. So arthritis, carditis, palsy, or partial paralysis of the face, or other parts of the body. Again, arthralgia, myalgia, numbness. And Lyme disease is the most common vector-borne disease in the Northern Hemisphere. According to the World Health Organization, there are 2,851 cases reported by the Public Health Agency of Canada in 2021, and those are preliminary results. And the CDC estimates that in the United States, there are about 300,000 cases annually. So it's not a small problem. Now, I'm going to shift to talking about antibodies, and antibodies are very important to understand for my project. I'm just going to give a very simple explanation of what they are and how they work. So antibodies are sometimes referred to as immunoglobulins, and they can be abbreviated as AB or IG. For my presentation today, I tried to stick to abbreviating it as IG. So whenever you see IG, I'm referring to antibodies. Antibodies in textbooks and in the minds of a lot of undergraduate students look like this. They actually come in different forms, but this is one of them. So you have these two heavy chains, two light chains, and you have this hypervariable region right here. Now, where do antibodies come from? Antibodies are produced by your body. They are proteins that are produced by your body when it recognizes a foreign invader, essentially. So when it sees either a pathogen or a piece of a pathogen, a protein, it says, I didn't make that. And it says, I'm going to have to do something about it, and starts producing antibody. And that's done by white blood cells, specifically B cells. And these antibodies are really important for identifying and neutralizing those foreign proteins and invaders. So the other thing to think about with antibodies is that ideally they are good at detecting foreign particles, but sometimes they make mistakes. And if you have antibodies attacking yourself, then you can end up with autoimmune problems. One thing I will point out is this hypervariable region up here that's gray. You'll see that it fits this antigen perfectly. This antigen is blue here, but it would not fit any of these other antigens. And that's just a very good representation of how specific these antibodies are. They are produced in response to a single antigen. So the antigen is the foreign protein, just to be clear. Okay, antibodies. So this is just an example of some of the different forms they can take. They don't all look like Ys. There are dimers, which would be IgA, and pentamers, penta meaning 5, which is IgM. You're probably familiar with IgM and IgG the most, because those are the two antibodies that are looked for on Lyme serology tests. The other antibodies have purposes as well, and we'll talk about what that is. So each class has a general function. IgA is generally important for protecting mucous membranes. So your urogenital tract, your respiratory tract, and your digestive tract. IgD is important as a receptor on naive B cells. IgE, everybody's probably dealing with a little bit right now because of allergies. IgE is important because it triggers histamine release. And IgM is important because it is produced early in infection before IgG is produced. Really, when we're thinking of antibodies, we're thinking of them sticking to a cell and attracting a macrophage for phagocytosis. We're thinking of IgG. IgG is important for antibody-based immunity against pathogens, and it can cross the placenta in humans. In other animals, it can either go across the placenta through milk or both. So as you can probably guess, based on the title of my presentation, we're going to be talking about maternal antibodies. So IgG is going to be very important going forward. So this is just to go over how these come into being. So the B cell recognizes an antigen. So again, that's the foreign protein, and it divides into clones. And then those clones are basically antibody factories, which then produce these antibodies, which can then bind to a toxin and neutralize it by just changing the chemical composition. They can immobilize a pathogen, they can trigger the complement and lead to lysis, and they can also attract white blood cells for phagocytosis. And that's what you're seeing here in this cute little diagram. Okay, so maternal antibodies. When I say maternal antibodies are antibodies that you inherit from your mother. And they're very important. But why? Because when you are born, you are new and you're coming from a sterile womb to a very non-sterile world. Animals build immune memory through experience. So those B cells that produce antibodies, some of those will remember how to make those antibodies. And then when it encounters that same antigen, it will already know what to do and it'll mount an immune response quickly. The problem is that the first response is always slow because it takes time for the body to actually produce this antibody response to a novel infection. If you're a newborn, everything is a novel infection, and that can be overwhelming. And so it's kind of like you have all the tools, you have all the things you need for a working immune system, but you don't have the experience. The analogy I gave the other day was it's kind of like if I went out and bought $10,000 worth of plumbing tools and then showed up at your house and said, I'm going to fix your toilet. You probably wouldn't want me. I wouldn't do a very good job. These guys don't know what they're doing yet. So how do we protect newborns? And that is through maternal antibodies. So your mom can give you antibodies that she has produced. So her B cells have made them. They're in her circulation, and then she can give them to you as her baby. So how does that happen? It can happen through lactation, so through breast milk. It can be ingested and then absorbed through the intestines. And that is actually the only way that a number of agriculturally important animals get their maternal antibodies. So for instance, cows, it's essential that they get that colostrum, that first really antibody-rich feeding of milk from their mother. For humans, IgG is going across the placenta. With dogs, cats, and rodents, it's going both across the placenta and through milk to different degrees. And we're going to talk a lot about rodents today because my model is based on mus musculus, which is the mouse. So we're going to be talking about that. So just keep in mind that in mice, there's both transfer through milk and across the placenta. So what are the limitations of maternal antibodies? I mean, they sound great. Unfortunately, they're finite. Unless you are receiving regular transfusions from your mother, as soon as they cut the cord, you're done. For other animals that are getting them through breast milk, it'll stop at weaning. And then it's going to wane over time. Those antibodies don't just hang out forever. They do eventually leave the system. You're also limited by your mother's experience. So in a scenario where you are taken to a new area soon after birth, where there are infections that she's never encountered, you may not have immunity to those either. It's also complicated when it comes to vaccination, and that's something that comes up a lot in the literature when you're looking at maternal antibodies, because if you already have antibodies floating around, it can actually make it hard to mount an effective immune response to a vaccine. The amount of antibody you inherit from your mother varies based on a wide variety of factors. It can include, the species, not just the species of the animal, but the species of the pathogen that the antibodies are against. It also depends on the sex. So there were studies showing that different offspring of different sexes got different quantities of antibody. And I even found some indication in a paper I read a while ago that altitude can have an effect. So whether you live in a mountain or near sea level, it could make an effect. So there's a lot of variation. It's not consistent. It also can be very specific. So just because you have an antibody to say Borrelia or Borrelia burgdorferi doesn't mean you actually have protection against all Borrelia burgdorferi. It might mean that you only have protection against a single strain. And that's going to get into my project because that's where we're going. Okay, so we're going to put the pieces together now. Borrelia burgdorferi is really interesting for a lot of reasons. Coming from my background, I'm really interested in the medical side of things and the health aspect, but it's also really interesting to ecologists. It's interesting because it can coexist with multiple strains in the same environment and remain relatively stable over time. So what do I mean by strains? Because strains can actually mean a variety of things. Strains, really, when it comes to Borrelia burgdorferi, a strain is defined by its MLST type. So MLST is multi-locus sequence typing. And that's very important from a phylogeny perspective. So if you want to know the actual relatedness and the history of these spirochetes, that's important. However, to your body, the most important thing for your immune system is the OspC type. So they can also be referred to as strains when really what you're referring to is OspC type. And that's what I'm going to do today. So forgive me if you are diehard, but when I say strains A and I, for instance, I'm really referring to OspC types. If anybody is at any point is interested in the MLST types for the strains that I use today, I can get you those. The interesting thing with these systems is that you will have multiple strains of Borrelia. Some will be more common, some will be less common, but the ones that are more common, you assume they're better competitors, but they never achieve a monopoly. And the strains that are less common, you think they're obviously poor competitors, but they never go extinct. So there have to be a bunch of mediating factors that are both creating and maintaining that type of diversity in the environment. Just to talk a little bit about OspC. So OspC is obviously just one protein. It's important because it's immunogenic, so it elicits a strong immune response, and it's a good candidate for immunoassays. It's also exposed on the surface of the bacterium. So for instance, the flagella, that's protein as well, but it's inside. So it's not exposed on the surface of the cells. Your immune system doesn't see it unless the cell is lysed. OspC is relatively maintained within a strain, the strain in my case being defined by the OspC type, but it's variable between strains. So you can very easily test for different strains of Borrelia by simply looking at the OspC type. Now, it's also already used for direct detection methods. So we use OspC, the genes for OspC to actually detect the DNA from Borrelia burgdorferi already. We know quite a bit about it. It is quite useful, and it has been shown to actually be a good indicator. So we'll get into that. Maternal antibodies in a reservoir. So this was a study that my supervisor, Dr. Verdau, did in Switzerland. For this study, I'm specifying here that was with Borrelia burgdorferi sensilato because it was with Afzelii. They used Ricinus ticks, and they used a Swiss and Finnish strain of burgdorferi, or of Afzelii rather. The bankfuls were shown to have 100% protection against the maternal strain. So the offspring of a female bankful would be protected against the same strain that its mother had 100% of the time at 5 weeks post-birth. There were some limitations to the study. As I mentioned, the two strains were from different places. So the issue there is that they wouldn't necessarily interact in the environment. It's also important to point out that there was only single infections. And as I mentioned, there are lots of these spirochetes floating around in a system, and quite often these hosts are co-infected. So they have multiple infections at the same time. And so it's not necessarily the best descriptor of a natural system. I'm just going to go over quickly what this looks like if you break it down into an easy diagram to understand. So you have a female bank vole, and she's being bitten by a ricinus tick. And the Foley-Eby here, this is the yellow strain. The yellow strain goes in and she develops antibodies against it and develops these yellow antibodies. Those yellow antibodies then go to her offspring after she has them. And then those offspring grow up, and they, at 5 weeks of age, they're challenged with ricinus ticks carrying either the maternal strain, the same strain that their mother had, homologous, the yellow, or the non-maternal strain, the heterologous strain, blue. And what you find is that they're only protected against the strain that they have antibodies for. So the antibodies, strain specificity of the antibodies does seem to be based on the OSPC type. My question is, could maternal antibodies mediate strain composition in areas with multiple coexisting strains of Borrelia burgdorferi? That's the overarching question behind my entire project. So we're going to talk about it. Some questions that I had going into this. I was interested in looking at two strains that were from the same part of Canada, in this case from Western Canada, and looking at specifically Borrelia burgdorferi using Ixodes scapularis and Mus musculus as my model. I wanted to see whether you would still have the same strain-specific antibody response, whether the antibodies would be strain-specific, whether a co-infection would reduce the protective efficacy of those antibodies, and at what point those antibodies would disappear and what rate they would decline. If we couldn't actually see them disappear, how low are they getting by 11 weeks of age? And at 6 weeks of age, we were hoping to see good levels of protection based on the study from Switzerland, but we weren't entirely sure, so we were going to try. And you might be asking at this point, the bank voles were 5 weeks, these guys are 6 weeks, because these guys, to work with them, they're a lot smaller. You have to let them get a little bit bigger, so that's why. Okay, so I think it's clear there are going to be levels to this study. You're going to have a maternal level and an offspring level. The maternal level is the mothers, but also you could include the fathers in that. They were not infected. They were simply used to breed. At the offspring level, you had a group of about 88 offspring. And those 88 offspring were separated into two groups, half of which was going to be challenged at 6 weeks post-birth, and half of which was going to be challenged at 11 weeks post-birth. The ones that got challenged first were group 1, so that's the 6 weeks challenge, and I abbreviate that to G1. The offspring that got challenged at 11 weeks post-birth, that's the second challenge, that's group 2, G2. So when I say G1 and G2, that's what I'm referring to. Okay, so my hypotheses were that maternal antibodies against Borrelia burgdorferi will be transmitted to offspring from infected mothers. Maternally transmitted antibodies will be strain-specific. Offspring will be protected from the maternal strain of Borrelia burgdorferi at 6 weeks of age, which is the G1. Protection of offspring from infection by the maternal strain Borrelia burgdorferi will be less so at 11 weeks of age. And I get questions all the time about what I mean by less so. I'm looking for breakthrough infections. So whether there's protection or whether the mice actually develop an infection. Then the offspring of co-infected mothers will have lower levels of protection against the constituent strain as the offspring of singly infected mothers. So we expected that if your mother is producing antibodies against two different strains simultaneously, her immune system is not going to be able to dedicate itself to producing twice the number of antibodies total. So you're going to get a reduction in the antibody levels, and that's going to be reflected in offspring. For strains, for the mice, and this might mean something to some of you who are in research, we use C3H HEJ mice. For the strains of Burgdorferi, I'm actually giving isolate names here. If you want the MLST types, I can give you those. 1622.2 with OSP type A, and I'm just going to refer to it as A. And then 1654 with OSP type I, and I'm just going to refer to it as I. So my objectives were to generate nymphs for the later offspring challenges, because nymphs don't just magically appear. You have to actually create them. And then I had to infect my female mice with the strains that I was interested in. So some female mice were infected with A, some with I, some with A and I, and some with a negative control. And then I bred those female mice to produce offspring. And then I sampled those offspring at 1 and 2 weeks to look at antibodies in the milk and blood, and specifically with the blood to confirm that there was actually antibody transmission to the offspring. And then I challenged my G1 group at 6 weeks of age, challenged my G2 group at 11 weeks of age, and then detecting is actually where I'm at right now. I'm working at looking at the infections developed in my offspring and the antibody levels. So a lot of the time, we're going to be talking about methods, and I want to just clarify that there are really 2 types of ways to look for Borrelia. You can use direct methods and you can use indirect methods. The direct methods would actually look for the bacteria or a piece of the bacteria. So that would include things like qPCR, culturing, microscopy. Indirect methods look for signs of bacteria. So that would include things like ELISAs and Western blots, where you're trying to detect the antibody response of the host against the infection. So those antibodies that we're talking about are really useful for a lot of reasons. And one of the reasons is because we can actually use them to detect an infection. With mice, you can actually say that there's an infection when you detect antibodies. With some animals, it gets a little more complicated because you can have antibodies left over after an infection has resolved. Mice don't resolve the infection, so it's actually pretty useful. Now, blood and serum would be indirect detection methods because there's not a high concentration of spirochete in the blood. Ticks, we use direct detection methods as well as tissue biopsies, and we're testing as we go. So we take ear tissue biopsies when we receive our mice. We take ear tissue biopsies after infections. We take blood samples when they arrive. We take blood samples after they're infected, and we take blood samples at the terminal, the euthanasia, the exsanguination at the end. So we're going to talk about progress now. So for generating nymphs, I'm always on the fence about whether to include this or not, but I thought if I'm going to include it somewhere, today is probably the day because people might find it interesting and I had time. So nymphs don't come from just the air. You don't go out and catch them and use them on your mice. You actually buy the larvae, and then you use those larvae to feed on infected mice, and then those larvae fall off and then molt into nymphs that you can then use for later experiments. Originally, one of my colleagues had to start by needle inoculating mice to start the process. Right now, I had the benefit of using pre-existing nymphs and then basically just multiplying them by using this method of putting about 100 larvae on each mouse. So this guy here, I just want to clarify, I think it's a really cool photo, but I also know that that for some people might look upsetting. The mice can't feel this because of the analgesic that the ticks are releasing, and actually they don't even try to groom the ticks off, even if they're on their face. They don't care. So yeah. Okay, so now we're going to talk about the maternal challenge. So once I had all the ticks I needed and everything was set up, we then went about challenging 32 female mice separated into 4 groups, 8 mice per group. We had 8 mice that got challenged with strain A, 8 mice that got challenged with strain I, 8 mice that got challenged with both strains, so that would be our co-infected group, and then 8 mice that were challenged with negative nymphs, so nymphs that didn't have any Borrelia. The one thing you may notice here is that we had to make a decision for the co-infected group about whether or not we were going to use for 4 ticks total or 4 ticks per strain, and we ended up with going with 4 ticks per strain. So in that one instance, that mouse would have 8 ticks on it. Then we bred our mice. That was actually pretty fun. They were very happy to have the company, and the babies were pretty cute. We had 30 mothers that ended up producing offspring. We had about 130 pups born. It's hard to know exactly with mice. And then there's some normal mortality that goes on. And we also decided to do some sampling at 1 week and 2 weeks of age because we had more pups than we needed. And then we had 88 pups left for the remainder of the study. I showed this to somebody earlier and they said, you want to clarify that that pup is dead? Yes, the pup is dead. The pup was euthanized before I took the stomach out for this photo. We were interested in looking at the milk, and unfortunately it's very hard to get milk from mice. And because the stomach, it curdles instantly, you cannot needle aspirate it or anything like that. So the easiest way to get it from pre-weaning mice is to actually extract the stomach. So we euthanized them, exsanguinated them, and then extracted the stomachs, and we've kept the contents frozen with protease inhibitors, and we're hoping to go back. Actually, that's my plan for next week, is to start looking at the milk to see whether there's IgG in the milk. Our progress. So we've shown that the mothers arrived with no exposure. The mothers were infected with the correct strains. The mothers had species-specific antibody response. So that's just saying not strain-specific, but actually just Borrelia burgdorferi. We knew that they had developed antibodies. Then there was no evidence of sexual or contact transmission to those male partners, which I think is interesting and something that comes up quite a bit. People are very interested in that. The offspring did have antibodies at 1 and 2 weeks of age. So we knew from there that things were working, and we expected to see hopefully some protection if there are antibodies present. Okay, so the 88 offspring that made it to the study were separated into that G1 and G2 group. So G1 was 45 mice and G2 was 43 mice. So they were randomly assigned to their groups, and then we just went back and balanced for sex because, as I mentioned earlier, sex may have an effect. So we challenged those mice with both experimental strains, and we expected them to develop an infection with the experimental strain that they did not have antibodies against, so the non-maternal or heterologous strain. And we were hoping that at 6 weeks, there would be protection in the mice from co-infected mothers, and we would see few or no infections in those mice. We were also expecting that the mice that had no antibodies, because their mothers were the negative control, to develop a co-infection themselves. And that was what we were expecting. And these are my results. So this is a really simple graph that actually says a lot. This is based on ear tissue biopsies 3 weeks post-infection in my offspring. This first graph is looking at the G1 group, and it's the susceptibility of those mice to infection. So what you're seeing here is these are the percent of the mice in that group that were infected with the respective strain. So the most important thing to see, first of all, is that far on the right here, there is the 2 bars on the right will actually show that there wasn't a huge difference in terms of their competitive ability with each other. They weren't out competing each other. So they were on a fairly level playing field if there were no maternal antibodies present, because that far right column is from the negative control. Then what I would like to show you is that in the case where there are maternal antibodies against A, but not against I, you had almost complete infection with I, which is what we expected. They don't have antibodies against it. But there were still some infections with A. They were breaking through. And in the situation where it was reversed and you had antibodies against I but not A, it was 100% A's coming through and no I's breaking through. Then you come over to here, and this is what gets exciting. So this is looking at the offspring that are the product of the co-infected mothers. And what you can see here is that there are a significantly greater number of breakthrough infections from A as opposed to in the singly infected group. But again, there's 100% protection against I. And this is statistically significant when you combine it with this group. So this group is the 11-week post-birth challenge. So this is group 2. And what you can see is pretty much everything looks the same, except for here. So right here, we're seeing that in the co-infected group at 11 weeks of age, there is 100% breakthrough of A and absolutely nothing coming through for I. So there was perfect protection against I all the way through, but not against A. And it got worse as time went on, and it was definitely negatively affected by the presence of a maternal co-infection. So there is a significant effect of co-infection on strain-specific protection. And if anybody is curious to see those data, the numbers and the p-values, I can send those as well. So the last thing I'm going to really talk about today are my ELISAs. This is what I've been doing pretty much nonstop for, I don't know, three or 4 weeks now. They are immunoassays that are detecting strain-specific antibody responses in serum samples from my mothers and from my offspring. And what we found was that the antibody levels are highest in the mothers, that they decline in the offspring over the 11 weeks. And that's the same regardless of which strain you're looking at. We also found that the antibodies didn't fully disappear at 11 weeks of age. And we also found that the strain-specific antibody levels were similar for both strains at any given time, which was kind of frustrating because we were expecting to see a difference. If we were seeing a difference in how the strains were able to break through, we were expecting to see a difference in the antibodies, but we didn't really. And one thing that we saw that was helpful and did describe what we saw was that the antibody levels were lower against both strains in scenarios where the offspring was the product of a co-infected mother. So if your mother was co-infected, you didn't get the same quantity of IgG against A and I as you would have if your mother had just had A or just had I. You actually got about half. Again, anybody interested in seeing those data, I'm happy to share those. So I have some other samples that I'm going to be looking at. Those include the tissues that I harvested at euthanasia. So we took ears, the tibiotarsal joint, the dorsal and ventral skin, the bladder, and the heart, but we also have the rest of the animal saved as well in case we want to go back and take more. We also sent off a series of samples for pathology to hopefully get an idea of what the fitness costs are to these mice. And then I also have xenodiagnostic ticks and challenge ticks. And xenodiagnostic is referring to when we did larval infestations on our mice to see whether or not there was transfer of the infection from the mouse to the tick. So the host to tick transmission, which is really important from an ecology perspective, because if it can't make it back into the tick vector, it's fairly irrelevant from an ecological perspective. So we have those as well to look at. Now I need to thank my supervisor, Dr. Martin Verdow, my co-supervisor, Dr. Janet Hill, my PhD committee, which includes Dr. Misra, Drs. Wilson, Cameron, Epp, and Rubin. And a really big thank you to my assistant, Georgia Hurry, who was invaluable for this. And then my coworkers, including Chris Zink, Ava Ullman, our tech, and Cody Koloski. And of course, I have to thank all the people who helped to make this possible, including various parts of the university and the Canadian Lyme Disease Foundation. Yeah.
Thank you, Alex, for your presentation. We can open it up to the audience for any questions or comments for Alex. I don't see any comments or questions in the chat box right now, but if you do have a question or comment for Alex, please raise your hand using the icon or unmic and ask your question. Go ahead, Dr. Foley-Eby.
Thanks. That was a really nice talk. It's mostly, I want to make sure I understand this because I thought that was really interesting. Your bar graph. Could you go back for a second?
Yeah.
Where you have the different inoculations. So yeah, this one and the next one, I just want to make sure I understand this. So the far left column, and then where it's A plus I minus, and then comparing with the third column.
Yeah.
Right. So the difference is on the left, they both have for the A strain, they've both been inoculated, but on the left, it wasn't inoculated for the I. Is that right? And on the right, it was. So my question is, or my confusion is, why are you getting it? Almost, am I interpreting this right, that having the extra I inoculation actually reduces the resistance to the A strain?
So if your mother was co-infected and she had both A and I, then you as an offspring had lower levels of protection against specifically A.
Right. So what's the mechanism there? Something about having that extra antibody is actually reducing the effectiveness?
So I'm not entirely sure, like this is the ongoing thing. I was really hoping there might be some answers when I looked at the strain specific antibody levels, but I'm just not seeing a difference between them. So I guess the real question is why is it happening for A and not for I? And I don't have a good answer for you. We've looked, for instance, one of the things we thought of was maybe the infectious dose is different. You're using ticks, so maybe some ticks are injecting more A or more I, and so you're getting different results. But we actually looked at, we can't say exactly how many are transferred through feeding, but we did look at the ticks, like a representative sample of the ticks that we were using to see what the abundance was, and there wasn't a statistically significant difference between the two strains. So we've talked about doing the possibility of some other studies, like looking to see if you could do like local tissue biopsies like right after infection to see what's actually being transferred to the mouse, because what's in the tick's gut is not necessarily what's getting in. But we're not sure. That if you have ideas, I'm all ears.
I know I'm not an expert in this at all. I just thought it was really interesting and caught me off guard. So thanks.
Yeah.
Thank you, Dr. Foley-Eby. Do we have any more questions for Alex from the audience? Well, I'll throw one out there. Again, ticks are not my area of expertise. So obviously the one thing I've always learned through my many years of university and grad schools is the more we know, the less we know. So what would be your next step of experiments that you would be pursuing to dive down more into answers or to take off from the initial results that you have so far?
Well, I think if you're going to apply this to a system, I mean, mus musculus is not really what you're dealing with. It would be like, I always say the name wrong, forgive me, Pyromuscus leucopus, like the white-footed mouse. So I think it would be interesting to use a host that's more ecologically relevant. It's hard to use in laboratory settings. That's my understanding of why we don't necessarily use it. But I think that would be a good step. I'm also really interested in the milk. It wasn't actually something we were thinking about a lot when we started out. And when we thought of it, we had the suggestion, I think about a week or two before our offspring were due to be born, and we had to figure out how we were going to deal with it, because you can't even just freeze it, because the the enzymes that are in the milk will degrade things. So you have to treat it right away. I've had the suggestion that the antibody levels may be different right after birth. So we had a 1 and 2 week sampling, but maybe having a sampling, 24 hours or 12 hours after birth might have been important as well. But I think there are lots of places to go from this. I think you could even do things like cross-fostering studies. Those would be really interesting, but I don't know. It's going to have to be somebody else, I think.
Yes, we can only do so long before we have to wrap up our grad project. Thank you, Alex. Do we have any additional questions for Alex from the audience? Don't be shy. Here's your opportunity. Okay, going once, twice. Well, that's it for questions for Alex. Thank you so much. Of course, if you do have questions, feel free to reach out and I'll be happy to pass along the questions to Alex and get a response and put it in one of our upcoming newsletters. I'm just going to switch the screen right now and just give you an update on our next presentation. Again, thank you very much for your engaging and visual presentation, Alex. I'm just going to check. There's a chat box. Yes, thank you. Everyone's saying thank you. Very interesting. I just wanted to let everybody know of our next series of presentations that will resume next week. We have Dr. Mark Solowski, who's joining us on Monday at 11 a.m., whose presentation will be on Lyme disease prevention, a summary and discussion of where we stand on Lyme vaccines and other immune-mediated approaches. Hopefully you can join us on Monday. We do have presentations Monday through Thursday next week. Also a reminder to please take part in our challenge, wear green or wear a green face mask, take a photo and share it with us to help spread awareness for Lyme disease. If you don't like to show your photos or send them in, if you are an artist or have any creativity, feel free to send in a snapshot of a piece of artwork or drawing that best represents Lyme Disease Awareness Month for you. All photos or entries that we receive will be automatically entered in a draw for one of 4 $25 Starbucks e-gift cards. And our draw will occur on live draw on May 31st after our last presentation. And you can send your images and photos to clydern@gmail.com. Again, thank you everybody for joining us today, and we hope to catch you next week as our events continue. Everybody have a great rest of your day and a great weekend. Thank you.